There's been a lot of interest, obviously, in cancer in general for trying to use immunotherapy where standard cytotoxic therapies have not been successful. And I think because of some of the tools that we have, such as PD-1 inhibitors, CTLA-4, all those other targets, we've been finally successful after decades of lack of success with immunotherapy. And in the hematologic malignancies, we think about things like CAR T cells, monoclonal antibodies in myeloma, specifically targeting two known important targets. We've heard some data on BCMA. All of this, I think, will roll up into really exciting new therapeutic options. But we know that even those approaches are not foolproof. And so you go to the other potential reason why cancer occurs, and that's genetics and genomics. Now, precision medicine is something that's been really successful in the solid tumors, but has not been quite as successful in the heme malignancies, and particularly in myeloma, mostly because many of the mutations that we see are not single drivers. Removing that blockade, like in CML, doesn't result in long-term remissions. And so part of an approach that I've thought about is using standard therapy with IMiDs and proteasome inhibitors, immunotherapy to try and get you down to low-level disease, and at that point you may have one or two clones that remain, and you target that with a mutation-driven therapy. So I think ultimately if we're going to cure patients with myeloma, it won't be just immunotherapy, it won't just be precision medicine, and it won't just be what I call biology-based approaches. It'll be putting all three together to really get rid of the disease. MRD really has really been developed because we've been a victim of our own success. When you're starting to get CRs reliably now, both in the induction and in the relapse setting, then all CRs are not the same, and you use MRD as a way to separate out really, really deep responses versus less deep responses. There's a lot of controversy in the community right now about using flow or using NGS, next generation sequencing, to measure MRD. And my personal bias is that next generation sequencing is going to end up being the modality we use because it's easily reproducible. And it gets you a log better depth of response, 10 to the minus 6, than you get with average flow cytometry. Now, flow cytometry may have other advantages in that it can give you immune profiling and other things that are useful. But I think that MRD it's here as an investigational tool, but it's not quite ready for prime time and certainly not ready for making therapeutic decisions. So there are three different consensus guidelines that are coming out from each of the chairs of the meeting. So the first consensus guideline is from Shaji Kumar, and what he's really talking about is MGUS and smoldering, giving guidelines on definitions, giving some level of risk stratification for both MGUS and smoldering, and then talking about standard ways to monitor these patients based on where they fit into the risk stratification guidelines. And I think that's really important because there's a lot of heterogeneity in terms of practice out there. I may see a high-risk smoldering every month, others may see them every 3 months, and there is some data to talk about how often you should see them, what imaging you should do, what are the standard tests you need to do to appropriately risk stratify them. So that'll be a nice and welcome consensus document. The second is the one I'm leading, which is based on high risk and special populations. And our goal is not to redefine high risk, but to give clinicians guidelines on what to use to assess risk. And so you can do things like FISH and cytogenetic testing. They should be done in certain methodologies. You can use gene expression profiling. You can use NGS. But we also created a category of clinical high risk, patients with plasma cell leukemia, extramedullary disease, things that I think we all sort of know or feel, but they've never been codified in any document. And I think that's really another strength of the manuscript as well. And then we go into recommended treatment approaches for patients, whether they're older or younger, and then focus on 4 special populations: patients who may be eligible for allotransplant, patients with frailty, patients with renal failure, and then emerging targets in high-risk disease. The third consensus statement is coming from Dr. Nupa Raji, and what she's going to talk about is vaccination and infection prophylaxis in patients with myeloma, which is an area that everybody has very strong opinions on, but there's very little data out there. And so what Nupa and her group tried to do was come up with some consensus agreement on areas where we need clinical trials to answer questions, but what best available data is to be able to do that. And so I think those three guidelines will be very practical, clinical things that will hopefully be available for practitioners in the near future. You know, the take-home message for patients is really twofold. One is, I think, one of hope. There's lots of new drugs and new targets that are coming out that I think will be very useful for them in the near future and hopefully approvable in the near future as well. The second is that, you know, one of the things I often hear from patients is, well, do you all talk to each other? You know, I can see a doctor in California, I can see a doctor in Texas, I can see a doctor here, but how do I know that you guys are all communicating and are working together? And I think what this meeting really shows is that there is that communication, not just in the US, but globally. Particularly in the myeloma community, I think we're a very close-knit group, and we do share our information, we do share our ideas. We may have opinions about who's right and who's wrong, but I think that sharing occurs really frequently, and that ultimately is better for patients.